in collaboration with Medical Faculty of Trisakti University. Yang hari ini topik kita akan membahas terkait COVID-19 updates. Nah, saya Lia yang hari ini akan membantu memandu jalannya acara sebagai MC. Nah, namun sebelum itu kita mau lihat dulu rundown acara kita hari ini. Boleh di next slide. Halo. Ya. Nah, rundown acara kita hari ini, yang pertama kita akan mendengarkan sambutan dari Wakil Dekan 1 kita, Dr. Eli, nanti. Kemudian kita akan langsung masuk ke dalam sesi kita yang akan dipandu oleh moderator kita, Dr. Nuryani. Kemudian kita akan membahas terkait molekular aspek of COVID-19 yang nanti akan disampaikan oleh Prof. Gerard Pauls. Kemudian setelah itu kita juga akan masuk ke sesi kedua, yaitu tentang laboratory testing updates on COVID-19 yang akan disampaikan oleh Intan Mustafa, Master of Science. Kemudian yang ketiga kita akan membicarakan terkait biomolecular laboratory update in FK Trisakti yang akan disampaikan oleh Dr. Monica. Kemudian baru nanti kita akan masuk ke dalam Q&A session, jadi Bapak Ibu Dokter bisa memberikan pertanyaan pada saat Q&A session nanti. Kemudian setelah itu kita masuk ke dalam sesi Prodia in Research yang akan disampaikan oleh Bena Zaira SSI. Nah, saya himbau untuk para peserta mengikuti acara sampai akhir, karena di akhir nanti kita akan kasih kuesioner, link kuesioner yang bisa diisi, kemudian nanti akan kami kirimkan e-certificate dari webinar hari ini. Nah, langsung saja, sebelum kita masuk ke dalam sesi yang pertama, kita akan mendengarkan sambutan dari Wakil Dekan 1 dari Fakultas Kedokteran Universitas Trisakti, yaitu Dr. Eli Herwana M. Biomed. Kepada Dr. Eli kami persilakan, Dok. Dr. Eli kami persilakan.
Bapaknya belum unmute. Selamat pagi Bapak Ibu hadirin dalam acara webinar pada pagi hari ini. Kemudian kepada para pembicara, sekali lagi selamat pagi, salam sejahtera dan salam sehat buat kita semua. Bersama ini pertama-tama kami akan menyampaikan permohonan maaf bahwa pada hari ini Bapak Dekan tidak dapat bergabung dengan kita di sini dan mewakilkan saya untuk acara pembukaan dari webinar pada pagi hari ini karena Dr. Aditya Pratsangka SPOG konsultan pada hari ini ada acara penting di dalam keluarga. Kemudian kami juga ingin menyapa para pembicara, terutama kepada Prof. Gerald Pauls. Welcome, Prof. Gerald Pauls. Welcome to the this webinar, and thank you for sharing knowledge today with us. Kemudian juga pada para pembicara lain. Webinar ini sebenarnya dilaksanakan bersama-sama berkolaborasi antara Prodia bersama dengan Fakultas Kedokteran Universitas Trisakti. Dan acara ini juga diselenggarakan dalam rangka perayaan Dies Natalis Universitas Trisakti yang ke-55. Jadi kita pada bulan November ini, tepatnya sebetulnya tanggal 29 November 2020, ini adalah lustrum 11 ulang tahun Universitas Trisakti yang ke-55. Sebagaimana biasanya kalau ada acara Dies Natalis, kita mengisinya dengan berbagai kegiatan. Antara lain tentunya dengan acara-acara ilmiah maupun non-ilmiah. Untuk acara ilmiah memang kita juga menyelenggarakan seperti biasa adalah webinar, tapi juga acara non-ilmiah seperti lomba-lomba termasuk lomba karya ilmiah itu juga tetap diselenggarakan, namun dengan keterbatasan saat ini tentu segala sesuatunya ada banyak yang berubah dalam pelaksanaannya. Sebagaimana pada hari ini yang seharusnya kita bisa bertemu secara luring, tapi dengan kondisi saat ini kita bisa bertemu secara daring. Tetapi itu juga membuat semakin luas dan memudahkan hadirnya para pembicara dari luar atau dari banyak kemudahan pula dengan acara webinar ini tentunya. Di tengah segala keprihatinan kita dengan masalah pandemi pada saat ini, tentunya banyak hal yang harus berubah. Ada work from home and stay at home, but life must go on. Dan justru kehadiran COVID yang tengah berlangsung dan mendekati lamanya ini sepertinya sudah hampir 1 tahun ya, dan belum pengendaliannya belum maksimal. Tentunya ini sepertinya memicu pula menggerakkan para ahli untuk mencari solusi melalui berbagai penelitian. Sebagaimana pula topik webinar kita pada hari ini, yaitu mengenai molekular aspek, laboratory testing, biomolekular laboratory. Nah, tentunya itu sangatlah penting untuk pengembangan penelitian untuk COVID-19 pada saat ini. Dan tentunya pula selanjutnya setelah jenis dari virus terdeteksi, patofisiologi juga lebih banyak telah dipahami, tentunya pengendalian COVID-19 akan menjadi target yang utama. Sebagaimana yang banyak juga diharapkan adalah nantinya akan di primadona mengenai vaksin yang ditemukan lebih aman, sangat efektif dan aman, itu juga dapat menjadi solusi dalam pengendalian. Dan semua itu untuk bervaksin dan sebagainya, pemahaman tentunya hal itu berkaitan erat dengan adanya berbagai penelitian. Maka kami menyambut baik dan selamat kepada penyelenggara dari Universitas Trisakti, para panitia, kemudian juga pada Prodia yang telah menyelenggarakan topik penelitian berkaitan dengan COVID-19 pada hari ini. Sebetulnya kolaborasi antara Prodia dengan FK Trisakti itu sudah sangat lama berlangsung dan terutama juga diimplementasikan dalam bidang penelitian ya, kemudian berbagai seminar ilmiah. Nah, itu yang banyak kita lakukan sebagaimana yang kita laksanakan pada hari ini. Dan kami juga telah terus menjalin kerjasama bersama dengan Prodia supaya penelitian yang dihasilkan bersama-sama dari Fakultas Kedokteran Trisakti bersama-sama dengan Prodia dalam bentuk webinar atau berbagai penelitian kelak akan memberikan banyak manfaat dalam bidang pendidikan dan pengembangan ilmu di Indonesia tentunya. Tapi tentunya publikasinya juga akan dapat menjadi suatu base evidence misalnya seperti itu bagi seluruh dunia. Dan tentunya akan memberikan manfaat pula bagi pengembangan ilmu dan kesehatan bagi seluruh umat. Pada akhir kata, kami ucapkan selamat mengikuti seminar dan terima kasih atas penyelenggaraan ini dan mohon maaf bila mana ada salah-salah kata dalam penyampaiannya pada hari ini. Terima kasih, selamat mengikuti seminar.
Terima kasih Dr. Eli atas sambutannya. Baik, selanjutnya kita akan masuk ke dalam sesi kita yang hari ini akan dipandu oleh moderator kita yaitu Dr. Nuryani. Beliau merupakan dosen dari Fakultas Kedokteran Universitas Trisakti dan beliau juga saat ini menjabat sebagai Editorial Board of Journal Biomedica dan Kesehatan FK Trisakti dari 2018 sampai sekarang. Beliau juga menempuh pendidikan di Universitas Trisakti dari pada tahun 1992, kemudian dilanjut juga mengambil spesialisasi dalam kesehatan fisik dan rehabilitasi di Universitas Indonesia. Langsung saja kita sambut Dr. Nuryani Sidarta yang hari ini akan memandu jalannya acara kita hari ini. Saya persilakan Dr. Nuryani.
Oke, thank you Miss Lia. Oke, Good morning, our vice dean and our honorable speaker, Professor Gerard. And then we have also Miss Intan and Dr. Monica as our speaker today. First of all, I would like to welcome our distinguished speaker and also our participants to this seminar regarding to the topic is the COVID-19 update. I will be the moderator today. So before we begin with our session, I would like to share some informations. First, please turn on the audio mode so we can avoid any disturbance during the presentations. And then the session will be we will have three speakers in our sessions. The first one will be Professor Gerald, and then we'll follow by Miss Intan, and then the third speaker will be Dr. Monica. She will talk about the biomolecular laboratory update in our Faculty of Medicine in Trisakti. Okay. Now allow me to share some biodata regarding to our first speaker, Professor Gerald. Hello, Professor Gerald.
Hello.
Thank you so much. We are so honorable to have you as our speaker today. So allow me to share some information regarding to your data. So Professor Gerard started his educations in 1979. Professor Gerard have a Master of Science in Biochemistry from the University of Utrecht. And then Professor have a PhD degree in 1986 from the PU University of Amsterdam. And then after that, from 1986 to 1988, working as an associate professor of genetics in Wayne State University, Detroit, followed by being a director of DNA and protein diagnostic in PU Medical Center. And then the last one from 2007 to 2019, Professor Gerard is the head of Center for Connective Tissue Research in the PU Medical Center. Right now, Professor Gerard is the visiting professor regarding to human genetics in two universities in Indonesia, in the University of Gadjah Mada and also the University of Diponegoro. And right now, Professor Gerard also being a consultant for genetic tests developing at Prodia Jakarta. Okay. The second speaker, Miss Leah, can I have a, yeah, thank you. So the second speaker will be Miss Intan Wibawanti Masfufa. She started the education from the Institute of Technology of Bandung. She has a bachelor program in clinical pharmacy and community. And then in 2010, she got a dissertation also from the University of Technology with the dissertation is treatment pattern of systemic lupus erythematosus in one of the government hospital in Indonesia. Right now, and I'm sorry, then from the 2016 to 2017, Ms. Intan has a Master of Science in Genomic Medicine from the Imperial College of London. Right now, he's working as the research support and scientific manager in PT Prodia. As we can see here, from 2008 until 2017, she has a lot of research. Dr. Monica is one of our colleagues in the Faculty of Medicine. Can I have the CV of the Dr. Monica, please? So Dr. Monica start her education as a medical doctor from the Gadjah Mada University in 2001, and then she continued her study in Master in Biomedics from the University of Indonesia, finished at 2005, and then after that he also continued her study, have intensive immunology from the University of Gadjah Mada, and in 2019, Dr. Monica has her PhD in discipline University of Adelaide, Australia, and a lot of publications that I will be too long if I read it one by one. Right now, she's working at the one of our staff in the medical biology department, and also right now he's also also working as a head of laboratory biomolecular in our faculty of medicines. Now, without further ado, please allow me to welcome the first speaker, Professor Gerald. You will have around 40 minutes to present your presentations, and then we will continue with the second speaker. The floor is yours, please, Professor Gerald.
Thank you, Dr. Nuriani. It's an honor for me to speak for the University of Trisakti. I will try to share my screen now. I think this. I think I'm sharing the wrong screen now. Wait a moment. Can you see my screen, my presentation now?
Yes, Professor, we can see your screen right now.
Okay, then I can continue. So I'm talking about the molecular aspects of COVID-19 now. And I'm saying this because there was a microscope in this picture, but the viruses cannot be seen with a normal light microscope. You can only see them with the electron microscope. They're so small. And I'm sorry about the text on this slide, but this is in Dutch. But I'm showing you on the right side one picture of the well, an artist impression of the virus. On the left side, we see the electron microscopy picture of the virus, and you can see the little circles there. The virus is very small. The scale on the bottom you can see here is 200 nanometers. So, and these little protrusions are, we call it the S protein, spike protein. These are the spikes. These are the crowns actually, which give the virus its name, corona. So they call them little crowns. We have the molecular aspects of the virus and also of the host, of course. There has been so much interest in the COVID-19 the last year, of course. I'm showing you here the data resource at the National Center of Biotechnology Information in the United States. There are already almost 4,000 ongoing and completed clinical trials for vaccines and for medication. And there are more than 70,000 open access papers already, and I've not read all of them, of course, but I'm trying to give you an overview of what's happening in this field. First, we need to know some very basic stuff. What are viruses? They are submicroscopic particles that can transmit disease. They were discovered in 1886 by Martinus Beijerinck in the Technical University of Delft in the Netherlands. They are not living organisms. So we cannot talk about dead virus because they're not alive. They need a living organism for replication. They consist of genetic material surrounded by proteins. And they contain genetic material, DNA or RNA. And a reminder, we have double-stranded DNA, like in this nice girl here, with the hair in double strands, and the RNA is a single strand. You probably recognize this lady from the Game of Thrones. And the central dogma of DNA in molecular biology is DNA encodes RNA encodes protein. And normally, this is how things work in human cells. DNA contains some 25,000 genes. Its transcription makes it into RNA. And we have about 100,000 transcripts because every gene can make more than one transcript. And we can make more than 1 million proteins. Most of those are the antibodies, of course. And the proteins have all kinds of functions like enzymes, DNA synthesis, signaling, structural support. And then if we look at the virus, host cells replicate the virus after infection. And I have an example here. The adenovirus contains double-stranded DNA. The DNA is copied by the cell into RNA and also into new DNA protein. And that way we get new virus particles. And the virus contains the code for its own proteins and uses the cell's resources to replicate itself. So that way the cell is forced to make new virus particles. The coronavirus is different. It contains single-strand RNA. The RNA is copied by the cell into proteins. And one very important protein is the reverse transcriptase. That enables the virus to be copied into DNA, which is in turn copied into RNA. And the RNA is copied into the proteins, and then we have new virus particles. An important issue here is that the reverse transcriptase and the DNA polymerase of the virus do not have a proofreading function. So that's why the virus has in the course of infection and later picks up a lot of mutations, more than our cells do. So how does the virus enter our cells? The spike protein binds to proteins on the surface of the ciliary cells in our airways. Here we can see how that happens. The virus, here you can see the RNA molecule, the spikes on the outside, and here we have the molecules, the ACE2 receptor molecules, which are used by the virus to enter the cells. It can also bind to a stress protein called GRP78. That's been overlooked a little bit lately, I think, but it's a very important issue because when the lungs are under stress because of the viral infection, the cells in the blood vessels will produce the stress protein GRP78, and the virus can use that same protein also to enter new cells. Surface proteins are usually meant for signal transduction, of course, but in this case, the first attack is on ciliary cells and the next one on the capillary blood vessels. And that's why we get serious lung problems. The vessels start to leak and you get, and the ciliary cells are stopping their function. So you get serious lung problems. Sorry. In young children, the ACE2 receptor has a very low expression. So they don't have this, hardly have any molecules of the receptor. So this may explain why young children do not get infected. And then the effect on blood, of the relation with blood pressure here. Normally, there's a balance between different proteolytic pathways. And normally we have in the kidney, we have angiotensin, which is changed into angiotensin 2 by ACE. ACE2 will break it down into a product that is regulating blood pressure and block organ damage that way. And angiotensin 2 type 1 receptor, if that's too active, it will cause lung injury, inflammation, and leaky vessels. There is a drug, losartan, which is specific for type 1, for the type 1 receptor, which may restore the balance. That's why there's an ongoing clinical trial at Mayo Clinic in Rochester, Minnesota, in the United States. There's no results of that yet. But it's interesting to see whether this will help. And we could also maybe look at what we call data mining. Many people are using losartan, and if we can find any information about losartan use among COVID-19 cases, that might show whether or not this prevents the very serious effects of the COVID-19 infection in some people, but we need very large numbers for that. That's not been done yet. This shows you the amino acid sequence of the human. It's hard to read it, but it's not the idea of this, just to show the complete amino acid sequence of bat COVID-19 from Wuhan, China, January 2020. Compared with the human virus sequence, and the middle line here is the comparison. This area on the right here, the blue area and the yellow area, are the receptor binding domain of the spike protein, very important protein of the virus. Here I zoom in on the receptor binding domain, and the red blocks show you mutations in the virus, comparison between human and bat virus. And most of these mutations are present in the area that's essential for receptor binding. There's also an area here in the 480 to 488 amino acids, which is the binding site for the GRP78 protein. So there's another mutation here, an insertion of 12 bases, 3 amino acids, which creates a cleavage site for human proteins, proteases, furin and trypsin, which by cleaving the protein, the spike protein here, will activate the virus to enter the cell. So this combination of mutations is very advantageous for the virus to be infectious for humans. So there has been a lot of discussion now. Are these mutations natural, or is it manipulation? Has this virus been created? It's possible that's been created, of course, and has escaped the Wuhan virus laboratory by accident. It's also possible that it is just evolution, selection of advantageous mutations by nature in the new host. At first I thought the time for, because they thought that the virus had been emerged in humans in 2019, only a half year before it was found. But there's a publication of 12 years ago that there was in Wuhan also, there was a problem with 12 people in a mine in Wuhan also, who died of a virus infection. But at that time, there was it was the bad virus. There was no human-to-human transmission. So then this publication has been forgotten. However, during 12 years, this amount of mutations it is possible to accumulate this kind of mutations in a virus without any manipulation. So there's still a discussion about that. This is a protein alignment of the virus as it was found in of the spike protein in the virus in January, and underneath that the very recent virus sequence. And there's only 2 mutations in the spike protein here. On the average, there are between 5 and 10 new mutations in many of the virus sequences that we get nowadays. And some of them are indeed in the receptor binding area. And this is a publication about the some of the mutations that indeed strengthen the infectivity of the virus. It's from Michigan State University. And here we see at the bottom here of the picture, we see the amino acid substitutions. Those are the important effects, of course. The mutations are found in the RNA molecule of the virus, but the effect will be seen in the protein. And the effect on the delta G, which is a thermodynamic expression of the binding energy, the binding free energy, and the higher the energy, the more the stronger the binding will be. And the color here, you can see the color scale here, is an indication of the frequency of those mutations found in the population of the viruses that are found in the mostly the American population actually here. And there's only one mutation here, the brown one here, which is the N439K. That's an glutamine to lysine mutation. It introduces a positive charge, and those mutations are usually have an effect on binding. In this case, a positive effect. So there's only actually one mutation that has a relatively high frequency in the population, this one. And here we can see over time, starting in January or December actually, until June, what happens to the new mutations in the virus in this area of the spike protein gene. And some of the mutations that have a positive effect, and we see here at the top, we see the N439K, the lysine substitution. Over time, the frequency increases. It's not really high frequency because 50 out of you have to look at the total number of sequences that have been found until now, and it's about 30,000. So it's not really a high frequency, but you do see an increase of the frequency of those mutations that have an advantage for the virus to be infectious. And those changes cause different clusters of sequences that we find in the world. So the original sequence was found in China, of course. We see the same, very close to the same sequence in Indonesia and Australia, and also in India. Cluster 4 is the one that is brown here and has the N439K mutation. So that's a little bit more infectious than the other strains, the other clusters. Interestingly, within Europe, we see the brown, cluster 4, and in the Netherlands, there's another cluster. Okay. If we compare Asia and with Europe and the Americas, Americas is North and South America. In East Asia, we see that the effects of COVID-19 are less severe. Claudia laboratory found in Jakarta that 84% of the people that test positive don't get symptoms. In the Netherlands, we don't know. We have too few tests. Somehow that's a policy here. I don't know why. In Italy, testing of entire village populations, they found that 40% of the people that test positive don't get symptoms. And that's a huge difference between the populations. So a possible explanation for the large difference could be that tuberculosis is very common in East Asia, not in Europe. And the BCG vaccination that's used against tuberculosis protects against corona. There could be many false positives, but I don't think so. Mutations in the virus cause more infectious or more virulent strains. That's an option. We've seen that those mutations do happen, but not in a frequency yet that can explain these huge differences. So I think the last explanation here, difference in DNA variations between human populations, could be more important. The BCG vaccination is very common in China. Everybody gets the BCG vaccination several times during his or her lifetime, and that may explain the low infection rate in China, but not in the rest of East Asia. I don't think that BCG vaccination is common in Indonesia. False positive tests may occur, but nearly all countries use the same PCR test, so that cannot explain this difference. And false positive tests depend on a threshold of the qPCR, the RT-qPCR is being used. 35 cycles maximum should be used actually. The PCR using a probe is more reliable. Mutations in the virus. We have seen, I've shown you that these are happening, but not at a rate that can explain the huge difference we see here. So DNA variations between human populations are present and may explain the difference in infection rate and disease severity. First, I want to show you the very recent, this week, overview of the COVID-19 cases worldwide. This is sorted on total cases per country. So you have a huge influence of the population size here. The most of the cases are found in the United States. And well, with the dependence on the population size is not very. Wait a moment here. Here I've sorted here on deaths, the number of deaths per million inhabitants, and then here deaths per million population here, and then we can see that well there are some tiny countries like San Marino and Andorra which have a very few inhabitants, so that's not very easy to show what's happening there, to know what's happening there. Belgium is also very high, but most of the countries that are with the red arrows here have Southern European descent. We call those the Latino people. Peru, Spain of course, which is where they come from. Brazil is coming from Portugal. The population is coming from Portugal actually, so that's also next to Spain. The USA has a large Hispanic population. Italy is also Southern Europe. So that's an issue. And if we still sort on deaths per million, and we go down the line, we can see here that Indonesia is actually doing very well. It's the position 108, only 52 deaths per million population, whereas in the Southern European countries and the South America, we have 1,000 deaths per 1 million population. In my country, the Netherlands, is somewhere in between. If you look at East Asia, here is filtered for Asia. We can see that Indonesia is 52 here. The Philippines are a little bit higher. And many of the East Asian countries are, especially China here, have a very low number of people dying from COVID-19 infection per million inhabitants. From Italian research, we have seen that serious COVID-19 patients compared with people that have no symptoms but are still infected with WES, all genes are sequenced, whole exome sequencing. We can see that there are 3 regions in the DNA associated with a higher risk for COVID infections. The genes in one area here are related to, there's an estrogen-related receptor. T cells are, of course, involved in immune reaction, and this is also a receptor. And then there are 3 regions that are associated with severity of disease, and some of the genes in these areas are zinc finger protein 598, which enhances immune response, ACE2, the receptor that binds the virus, and the lysosomal trafficking regulator list. And so I thought, well, maybe we can see if there are any common variants in those genes in the European populations that are not present in the East Asian population. So I went to the Genome Aggregation database, and which has the genome sequence of, so the entire DNA of more than 300,000 unrelated individuals that have no apparent disease. And this is how you see the information in the database. This is the amount of information for one gene here, the list gene. And you can see the exons have more information than the introns, which is obvious because you also look at whole exome sequencing. It contains all the DNA variants in individuals from different populations. And I took those, I copied those data from the database in Excel and predicted the effect of variants in silico using a software called Alamut from Sophia Genetics. And ACE2 is X-linked, so men have only a single allele, and then we call not homozygous, hemizygous, they only have one allele, so if that's mutated, they don't have a normal allele. And then we can see that variants with a positive effect on COVID-19 binding have a high frequency in Europe and the United States. Well, actually, the United States is mainly European ancestry, of course. And that variant is not found in East Asia. So COVID-19 is predicted to be more severe, to be more infectious also in the European population. I checked all these genes and I found that variants with a negative effect on function or a positive effect on the virus infection are found more often, like here in the European population, 16.7% versus 4.2% in the East Asian population, and also in the Latinos. So these effects are predicted. We have no functional evidence yet, but it's a huge difference in frequencies, as you can see. And then there was a publication in the JAMA recently from Nijmegen in the Netherlands, showing that loss of function mutations in the TLR7 gene cause very severe in young men. This is also an X-linked gene, So, and then I checked the genome, and then these rare mutations, the loss of function variants, are found in the genome aggregation database. The frequency is almost 0. So we can see an antivirus response in the human system. So there is a logical explanation for this here, but it does not explain the population difference because these variants are extremely rare. So apparently there is a combination of common variants and rare variants that cause the severe effect of COVID-19 infection in some people. And many more genes, especially in this pathway, may have an effect on the susceptibility to COVID-19 infection and severe disease. So some new mutations in the COVID-19, must be 19 here, of course, cause increased infectivity and are being positively selected during the pandemic. Common and rare variants in human DNA cause susceptibility to infection and to the severe disease. And differences in disease severity between populations are mainly due to variants in the human DNA. And difference in COVID-19 infectivity between populations are due to variants in the human DNA and to selection for new virus mutations. So the disease severity, the severe cases, those are mainly due to variants in the human DNA, so the susceptibility to severe disease. It's not caused by mutations in the virus, but the